This work really came out of a couple of different observations over the last decade or so. First is that individuals with sickle cell disease seem to have a higher relative risk of AML or acute myeloid leukemia compared to individuals without sickle cell disease. That absolute risk remains low, something on the order of 12 cases per 100,000 individuals. But when you put that into perspective, AML is a rare cancer, affecting only 2 to 4 individuals out of every 100,000 per year. We also note that when we use curative therapies for sickle cell disease, such as allogeneic stem cell transplantation or gene therapy, that individuals with sickle cell disease do have an increased risk of developing myelodysplastic syndrome or AML following these therapies. And then thirdly, we've observed that the types of MDS and AML that individuals get when they have sickle cell disease is actually a high risk type of AML. That is to say that the overall survival is less than a year, and it tends to have high risk molecular features like TP53 mutations or complex cytogenetics or changes in their chromosomes. And so all of these things together make us think, is there something biologically relevant to the onset of MDS and AML that's intrinsic to sickle cell disease? Are people with sickle cell disease more likely to develop MDS and AML because of something different about their blood cell biology? In parallel, we've had this increase in our understanding of clonal hematopoiesis, a precursor of MDS and AML. And so naturally, one of the questions that we ask is, are people with sickle cell disease more or less at risk of having clonal hematopoiesis, and particularly clonal hematopoiesis that is high risk for transforming to MDS and AML compared to people without? And so that's where our study really comes from, is trying to understand what is the prevalence or the rate of clonal hematopoiesis in people with sickle cell disease? Is there a difference in the gene distribution, since we know that different genes have different risk for developing into MDS and AML? And is there a change or a difference in the onset in terms of time? Are people with sickle cell disease more likely to have these higher risk types of clonal hematopoiesis or MDS AML precursors earlier in life relative to people without sickle cell disease? And so we designed a study where we pulled together samples from 17 different cohorts from around the world. So this is really a multinational study that involved clinician scientists from many different institutions within the United States, Sub-Saharan Africa, Canada, and South America. And we pulled these samples together and ended up with about over 3,000 individuals with sickle cell disease, a cohort of Black individuals without sickle cell disease, as well as a cohort of mostly pediatric individuals with thalassemia. And our age range in this population was from 0, so infants who are as young as 6 months old, to 86 years old. So we really had the full age range across the lifespan to analyze here and compare sickle cell to non-sickle cell disease. And so then we used a high sensitivity sequencing platform that was pioneered by the Lindsley Lab here at Dana-Farber to help us understand what the rate of clonal hematopoiesis or mutations in these age-related genes that are associated with developing MDS and AML and other types of clonal hematopoiesis actually are in the sickle cell versus non-sickle cell population. And to put our assay into perspective, typically we can get down to a limit of detection of about 1% variant allele fraction. So that's 1% of the copies of the gene are mutated. We can usually do that with our clinical assays that we use to treat and analyze patient data. This particular assay that we're using in this study actually gets down to 0.1%. So we're orders of magnitude lower in terms of our ability to detect very small variants in this particular study. The other thing that we added in our sequencing platform was the HBB gene. So HBB is the gene that's mutated in sickle cell disease. And this allowed us to validate the actual sickle cell disease genotype as in our sickle cell disease population, but also to tell In our non-sickle cell disease population, which individuals have sickle cell trait or the heterozygous state of sickle cell disease, and which individuals actually have a completely normal hemoglobin. And then lastly, we included some germline is to say that we added genes like DDX41, telomere maintenance genes like TERT and TERK, and RUNX1. And all of these are genes that are associated with an inherited predisposition to blood cancers like MDS and AML. And so really we are able to say across our age spectrum, is there a difference in the type of clonal hematopoiesis that we're seeing? And then we're also able to compare within the limits of our sample size, different subtypes of HBB genes, sickle cell disease to sickle cell trait, or sickle cell disease to thalassemia. And then lastly, we're able to say are individuals with sickle cell disease also co-inheriting any of these leukemia predisposition genes. So in terms of our results, we had a couple of provocative findings. The first was that clonal hematopoiesis actually is occurring much earlier in sickle cell disease relative to non-sickle cell disease controls. In our 0 to 19-year-old population, so our pediatric population, we saw that the rate of detection of clonal hematopoiesis was something like 10.6%, and this was compared to 3.5% in our non-sickle cell disease population. So people with sickle cell disease or children with sickle cell disease are almost threefold more likely to have clonal hematopoiesis than individuals who do not have sickle cell disease. When we analyzed which genes were causing this, we first thought that maybe it would be the genes that are most commonly mutated in clonal hematopoiesis in the general population. And these are genes like DNMT3A and TET2, and they're associated with aging. So in our older adult populations, we see a high prevalence of DNMT3A and TET2CH. But when we looked in our sickle cell population, particularly in this pediatric population, we actually saw that though there was a slight increase in the prevalence of DNMT3A and TET2CH in our pediatric population pediatric sickle population relative to controls, it wasn't enough to fully explain the real increase in prevalence that we were seeing. So then we looked at the DNA damage response pathway mutations. So these are genes like TP53, PPM1D, ATM, and CHEK2. And what we saw was that the prevalence of these genes, which are considered higher risk subtypes of clonal hematopoiesis, the prevalence of these genes was higher in sickle cell disease at almost every age group relative to non-sickle cell disease. So in our pediatric populations, in our young adult populations, and our adult populations, sickle cell disease was associated with a higher prevalence of clonal hematopoiesis involving TP53, PPM1D, ATM, CHEK2, relative to what we saw in a non-sickle population that was age and race matched. We then did a deeper dive into our pediatric population, leveraging about 150 individuals that we had who had samples in infancy and then had later samples collected at age 3 and age 10 to 11. And what we saw was that we could detect clonal hematopoiesis in nearly 5% of these individuals who were less than a year old, so 6 to 12 months of age. That ability to detect CH actually increased as these individuals age. So about 6% of 3-year-olds had clonal hematopoiesis, and about 7% of the 10 to 11-year-olds had clonal hematopoiesis. And the other observation that we made in this subpopulation was that all of the variants, the clonal hematopoiesis that we saw in infants, actually persisted from infancy to later childhood. So if you developed a variant at 6 months, you maintain that variant, or that variant was fit enough to stick around at age 3 and age 10 to 11. The other thing that we saw was that about 4% of individuals who did not have CH in infancy actually developed CH at age 3 or age 10 to 11. And so in this pediatric population, the dynamics are such that the rate of clonal hematopoiesis starts to expand as early as 6 months of age, and it continues to expand throughout childhood. Lastly, we looked at some specific scenarios just to see how generalizable this is, and we saw that sickle cell trait actually has a very similar prevalence and gene distribution of clonal hematopoiesis as individuals without sickle cell disease. And so this is suggesting that heterozygosity, having just one copy of the sickle allele, is not enough to actually increase your prevalence of CH. And then in our thalassemia population, we saw that this increased prevalence of CH was actually specific to sickle cell disease and not sort of generalizable to other types of beta hemoglobinopathies. Individuals with thalassemia had less clonal hematopoiesis than individuals with sickle cell disease, and they had about the same amount of clonal hematopoiesis as individuals without sickle cell disease. And then lastly, when we looked at our germline variants, we did not observe any co-mutation of germline predisposition alleles or mutations in DDX41, telomere maintenance genes, or RUNX1 in individuals with sickle cell disease that could really explain why we're seeing more clonal hematopoiesis in this population. So put all together, we're seeing that sickle cell disease is associated with this higher prevalence of CH, and that the onset of this CH actually is occurring in early childhood. And this is really not heard of in a general population of children, unless, of course, they have some sort of bone marrow failure syndrome or other predisposition to myeloid malignancy. Children typically do not have detectable clonal hematopoiesis. Second, we're seeing that this precocious clonal hematopoiesis is actually associated with mutations in the DNA damage repair and response pathway, so these TP53 and PPM1D genes. And this is important because these are the mutations that we're seeing associated with the myeloid malignancy, or the MDS AML, that we're actually seeing in sickle cell disease patients. Altogether, this is really providing a plausible mechanism for the observed MDS and AML that we're seeing in sickle cell disease, and really opens up the doors for us to ask some real key biological questions as to how CH is initiated or selected for in these populations, to really do some deeper dives into what aspects of sickle cell disease really are contributing to the development of clonal hematopoiesis.